Hello, welcome to March 13th, Friday's lecture. This is the one we missed because of the closing of the school. We're going to start with page 853 in the book. Now, I'd mentioned that we're going to copy something off the board. So the slide that you see in front of you now is the one that you would have copied off the board. This is the one that most of today's presentation is based upon. So please keep this particular slide in mind. Now this slide is going to be related to page 853, so we're going to go ahead and move over to page 853. This is now page 853 in your book. Okay, next slide. Now what we're going to talk about is level 1 of that handout that you saw earlier. Level 1 is innate. Now innate refers to something that is inborn. So you're actually born with that particular ability with level 1. Now, level 1 is also non-specific. That means it does not go after a specific germ. So again, level 1 is innate and it's non-specific. Now, using that chart, I'm going to go down that chart for level 1. The first thing that's mentioned there is mechanical and chemical barriers. Now, these are the things such as mucus from your nose, the pH of the vaginal tract or in the mouth. That's all part of the mechanical and chemical barriers. So urination, defecation, these all belong to the mechanical chemical barriers. Now in your textbook on page 855 is a full page chart giving you examples of such a thing as mechanical and chemical barriers. Now you don't have to memorize them all, but it would be a good idea to get a feel for page 855 and the different examples of mechanical and chemical barriers. The next thing we're going to look at on that particular level 1 is interferon. Now this was mentioned last class as one of the cytokines. Now interferon comes from a virus infected cell. So interferon is from a virus infected cell. Again, it is innate and it is non-specific. So that's interferon. I'm going down the list again. The next item on that list was fever. Now, fever has a good and a bad. The good thing about the fever is it raises the body temperature to a point where germs that are in the body can't survive. So it's in essence a way to kill the germ. But at the same time, as you're about to see on a slide that I get to, fever also has a bad part, and that is it can cause brain damage. So we're going to look at that in a moment. So are we all right so far? So that's bullet number 1, 2, and 3 under level 1. Now I'm going to move my slide further down, and we're going to get to the NK cells. So I don't care about this picture, but I'm going to stop on a picture, and that would be this one. And this picture is found on page 856. So page 856. Now page 856 deals with natural killer cells, also called NK cells. Now you can see from my chart the NK cells release a chemical called perforin. Perforin is the chemical that's actually going to pop the cell. So a germ is actually popped using perforin, and NK cells do that. You also see on the same page I'm on, page 856's picture, a term called apoptosis, which is actually killing that particular cell or popping that cell. So NK cells will use perforin to destroy the cell, and that's called apoptosis. So far I've done bullet number 1, 2, 3, and 4 from level 1. I'm now on the next bullet, which is inflammation. So I'll move to that slide next. So as I move further on the slides, you're going to come across inflammation. Now inflammation will be this particular slide, slide number 45, and it deals with the same thing that's under level 1. These are the features of inflammation: redness, heat, swelling. Of course, swelling goes with the word edema, pain, and then loss of function. So these are the properties of inflammation under level 1. Okay, so we've completed nearly all of the items in level 1. The next thing we're going to look at is complement, which is the last item. So I'm going to go back on my slide, and we're going to come across complement. Now all you're going to write down for complement is that it's a protein. So complement is a protein. Now what complement does is it covers the germ. It makes it more visible. So complement is a protein that will actually cover the germ and make it more visible. Now who are we making it more visible to? As you can see from the chart that we started with, is it makes it more visible to level 2, which are the macrophages. Now what that word opsonization that you see on the slide, opsonization is the process of putting complement on the germ. So opsonization is the process of putting complement on the germ. Now again, what does complement do? It makes it more visible to the rest of the immune system. It makes it more presentable, more visible. All right. Okay, so that's now covering level 1. So we've finished covering level 1 of that chart. Now I'm moving over to level 2, and as you can see from the chart, the macrophages are under level 2. We are still innate because level 2 is non-specific and it is still innate. That means it's inborn, you're born with it, it doesn't go after a specific germ, so it's non-specific. So macrophages. Now if you recall, we had different kinds of fixed macrophages from the past. The Kupffer cells from the liver, the dendritic cells from the skin, the Peyer's patches from the intestines. So these are all under the fixed macrophages. Now the next thing we have on that slide there, the original one, is the word phagocytosis. So I'm now going to show you a slide that deals with something like phagocytosis. Now the slide I'm moving to is just a generic one, and it looks something like this. Let me give you a page number. This is page 856 as well. Again, I'm picking a random one. And as you can see, it mentions macrophage on the title there, macrophage. So what does the macrophage do? It performs a process called phagocytosis, which actually takes the germ, which you can see in the picture. It takes a germ and then it puts it in a phagosome, a lysosome, which contains digestive enzymes. So you're actually destroying the germ and breaking it down. Now, if you look on the slide, the original one, phagocytosis by way of phagosomes, which are enzymes that break down the germ, the next thing we do after breaking the germ down is presentation. Now, who are we presenting it to? We are presenting it to level 3, which are the B cells, and level 4, which are the T cells. So we're actually presenting to level 3 and presenting to level 4. So B cells, T cells are getting that presentation. So macrophages will present to 3 and 4. Okay, moving on in our chart, we're now moving to level 3. Now, before we do that, we need to cover two other terms. The two terms we need to cover, make sure we cover them, is coming up on a slide in a moment. And it's something we've talked about in the past. So before I move to level 3, we need to take care of this particular slide. Now the term we did in the past was diapedesis. And if you recall, diapedesis is also called emigration. So diapedesis or emigration. The other thing we took care of last time too, and from a previous chapter, is chemotaxis. And if you recall, chemotaxis is when the white blood cell actually tracks down a germ. So when white blood cells are tracking a germ, like finding a slime, if you find slime, you know that a slug or a slime has been there, like a snail's been there. So that's what we're doing. Chemotaxis is tracking the germ. So white blood cells will track chemically the germ. So make sure you're aware of these two terms, diapedesis and chemotaxis, from a previous chapter, now recalled again. Okay, so now we can move forward. So now we're going to cover level 3. Now before we talk about level 3, we need to talk about this particular slide. This deals with level 3 being adaptive. So level 3 is adaptive. It is also specific. So level 3 is adaptive, but it's also specific. Now level 3 will deal with B cells, B as in boy, B cells. Now why do we call it specific? Because it goes after a specific germ. And why do we call it adaptive? Because it actually adapts with memory. So the unique thing about level 3 is that it has memory. So not only are we going to make germ fighters for now, but we're also going to make the special germ fighters for memory for the next time. But keep in mind that memory is always for that specific germ, even the second time around. So level 3, B cells. Now B cells make plasma cells. That's what's mentioned on that original slide, the first slide. So B cells produce plasma cells. Plasma cells make antibodies. Now, on the slide, I wrote antibody slash immunoglobulin. Remember the term. On this side of the ocean, we tend to say antibodies. Overseas, they use the term immunoglobulin. So one more time, B cells make plasma cells, which make antibodies. Now, this type of immunity that we're talking about for level 3 is called humoral immunity. Okay, so that's the slide for level 3. Okay, now I'm going to continue with the original slide showing level 4 now. Now, level 4 is also adaptive. Level 4 is also specific. Now, because it's adaptive and specific, it too has memory. So T cells for level 4 do have memory. So memory for T cells. The other thing we need to keep in mind is we have two major types of T cells. We have helper cells and we have cytotoxic cells. So helper cells, cytotoxic cells. Now, the helper cells are the only cell that can talk to the cytotoxic. You'll see that slide in a little while. So helper cells are the only ones that can talk to cytotoxic cells. Now, on the original slide, it says helper cell, and then there's a little ... showing you the term interleukin. So yes, helper cells will talk to cytotoxic using interleukins. Now, last class, we mentioned certain cytokines. If you recall, the term cytokine means it's a group of chemicals that tell a cell what to do. So in this case, level 4 can actually talk to itself. The helper cell can talk to cytotoxic. Now, before I go any further on that chart, the original one, notice there's also a dotted line that went from level 4 to level 3. So from level 4 to level 3. That's because level 4 also produces cytokines in the form of interleukins to talk to level 3. And remember, interleukin is only a communication between levels 3 and 4 typically. So 4 can talk to 3 using interleukins, and then also the helper cell can talk to cytotoxic using interleukins. Now, the next thing we see on that slide there, the original one, is the markers. You'll see that today in a little while too. So helper cells have a marker called CD4. The cytotoxic killer cells have markers called CD8s. Okay, now we can continue with our slide. Now, on the slide that you see in front of you now, at the very bottom, it says cell-mediated immunity and humoral immunity. Now, you don't really need to write that, but if you look at the original slide that I provided, it's already there. So cell-mediated immunity is for T cells, or level 4. So cell-mediated immunity, T cells, level 4. Humoral immunity, as I mentioned a moment ago, is for B cells, which is level 3. Okay. Now, this slide is found on page 864. So this is page 864. We're going to start from the right-hand side and quickly go through this chart. It is sort of a compilation of the one I've provided. So on the right, you see in yellow, humoral immunity, which is level 3, B cells. Now, you already know from the past that B cells deal with extracellular stuff like bacteria. So B cells will fight extracellular like bacteria. And in this picture, you see that the B cells produce plasma cells and the plasma cells produce antibodies. Moving over a little bit on the same picture here, we see that helper cells, the helper T cells, produce cytokines. This is the same arrow that's on the original chart of mine that shows you that level 4 can talk to level 3. So in our current picture, the cytokine is really the interleukin of level 4 talking to level 3. You also see that in the blue banner, it says cell-mediated. We already know it deals with T cells. We also know that T cells are for what? Intracellular, such as viruses and cancers. Also on this picture, we see what we already have on our chart, where it says helper cells talk to the cytotoxic by way of interleukins or a cytokine. Now, the one that you got to be careful for is the cytotoxic on the far left here. Cytotoxic cells are sometimes called killer cells because their only job is to destroy the cell. So in the picture there, you actually see cytotoxic T lymphocyte just damaging or destroying the cell. It does use perforin, just like the natural killer cells, and it causes apoptosis. Now, what's the difference between cytotoxic killer cell, which is for level 4, versus the natural killer, which was level 1? The difference is level 4 is adaptive, level 1 is not. Remember, level 1 is innate. Also, level 4 is specific, whereas level 1 is non-specific. So the natural killer cells of level 1 go after anything and everything, whereas the cytotoxic killer cells specifically go after a specific germ. Okay, that was page 864. Now we can continue. Draw a line in your notes, and we're going to start with this slide. Now, this slide is called antigen. Now, the rule of thumb is this definition. Antigens are anything that creates an immune response. So anything that creates an immune response is called an antigen. Now, the slide shows you examples at the bottom. You don't need to worry about all those examples. Just be aware of that definition. Antigen is anything that creates an immune response, and they'll be fine. Next slide. Now, the bottom of this slide that I'm on now, it says autoimmune disorders. That's what we're going to see on the next slide. Autoimmune disorders are when your body fights off itself. It treats itself as an autoimmune. So the next slide is one worthwhile copying. Okay, and I'm going to get there first. Here it is. This is the one you want to write down. This is called autoimmune disorders. And on a test situation, what we have here is diseases where they are fighting itself. They're fighting your own body. And 3 examples that you see on the slide are rheumatic heart disease, type 1 diabetes, and multiple sclerosis. Those 3 are often asked for on a test. So under autoimmune disorders, we have the rheumatic heart disease, type 1 diabetes, and multiple sclerosis. Okay, make sure you jot that down. Going back, we're going to go back to this slide now. Now, this is page 865's picture. Now, I've already told you that antigen is anything that creates an immune response. But at the same time, you see that in this picture, not only do we have an antigen, but it has different type of markers coming off of it. Those little markers coming off of it is called an epitope. So you can see different shapes there. You see a square, a oval shape, a box shape, a U shape. Well, all of those little markers that are coming off the antigen are called epitopes. That is what we specifically react to when we react to something. So we are really reacting to epitopes. Okay, so we're actually reacting to epitopes. Next slide. The next slide is haptens. That's the one I care for on this picture. Now, you don't need to write all of that definition there. Now, haptens are anything that can cause an allergic reaction, such as poison ivy, allergic to peanuts, allergic to penicillin. What we have here is a hapten is a piece of an antigen. So if we look at an antigen as a rectangle box, so if an antigen was a rectangle box, a hapten would be say a corner of that rectangle box. So if you had a box and that whole thing was an antigen, you're reacting to a piece of the antigen, and that's a hapten. So when you're allergic to something, you're not allergic to the whole antigen, you're allergic to a piece of that antigen called a hapten. All right, draw a line. Okay, this is our next picture. This is page 866. And I'm looking at this picture here of a T cell, T helper cell. If you look carefully, it says CD4. That's the marker for a T helper cell. The green one is the cytotoxic cell, and its marker is called CD8. And in the original handout I've given you, that's already labeled for you. So CD4 goes with helper cell, CD8 goes with cytotoxic. Okay, that's all you need there. Don't care. All right, now the next slide we're on, which is this one talks about antigen-presenting cells. You don't need the whole definition there. Just be aware of APC is antigen-presenting cell. On a test, you need to know the two examples. Macrophages and B cells are antigen-presenting cells. It will become self-evident as I move through a couple more pictures in the chapter. So antigen-presenting cell. Now, the next thing on that picture or that slide says major histocompatibility complex. Now, the MHC deals with the cell's markers. So when you give an organ to someone, when it comes to organ transplant issues, these are the markers that have to match. So if you're a very good donor to someone, then your organ matches very well in terms of MHC to the recipient, the person getting the organ. So MHC deals with organ transplantation. Okay. The next picture I care for is page 868. Now, if you look carefully, in the yellow banner, it says antigen-presenting cell. That's what this is. And if you recall a moment ago, I said that only two things will serve as antigen-presenting cells for us. Macrophages and B cells are antigen-presenting cells. Here I have a germ eaten up by a phagosome, very similar to the one earlier, and then notice what it's doing at the very end. So in the very end, it is presenting. So from my chart that I provided, it says that macrophages will present to level 3 and level 4. So macrophages will present to B cells, macrophages will present to T cells. And as you also know that B cells can also be presenting cells, so B cells can talk to level 4, the T cells. So these green balls at the far right are actually being presented to someone. That's page 868. Moving to the next page is page 869. Now on this slide, I see this bluish color blob, and notice it's not properly labeled. That bluish blob is an antigen-presenting cell. It could either be a B cell talking to level 4. So in this example, if this was a B cell, it'd be talking to level 4. If this were a macrophage, then macrophage being level 2 can also talk to level 4. And that's what this picture is showing. Talking to either a T cell in terms of a helper or talking to a T cell, which is a cytotoxic. Now the stuff at the very bottom that's been cut off, don't worry about it because it's already on your original chart. It says that the helper cells have a MHC class of class 2, whereas the cytotoxic cells have an MHC class of class 1. And that's at the very bottom of your original paper. Okay, moving on. I already mentioned this. Moving on further, 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 further. Now I'm on this slide. This is slide number 84. Now on this slide, slide 84, you see this word called self-tolerance, and it deals with T cells. Self-tolerance is the idea that says that while you're inside mommy's tummy being created, and some things are now defined as you and some things are still considered foreign, well, the T cells are pretty much going to tell everyone else, hey, wait until everything is complete before you make a distinction between what is already you and what is not yet you. Because as you're developing, you actually take on a characteristic with the markers on the MHC markers that we mentioned earlier. So it takes a while for you to get an identity. And the self-tolerance is saying, hey, let's not destroy you until we get you finally put together. So that's what that is. All right, next slide. Do not care. Okay, and I'm moving to the next important slide. Okay, this is now the next important slide. It is page 874 and 875. This is an important slide. The slide number is 93. So slide number 93. So page 874 and 875 are important pictures. We'll start with this one, which is picture B on page 874. Now here's where that APC comes into place, antigen presenting cell. As you can see, this blue antigen presenting cell can either be a macrophage level 2 or a B cell level 3. And in this example, it is actually stimulating, and that's the word I would use, it's actually stimulating a helper T cell. If I read the caption down here at the bottom, I notice it says it not only activates the current helper T cells, right, current helper T cells, but it also makes memory helper T cells for the future. And if you recall, level 4 does have memory. It has memory for the future. So not only are you making active helper cells for now, but you're also making memory helper cells for the future. That's this slide so far on page 874. Okay, now moving to the next slide, same page, page 874, picture A. Notice this time, do you see a arrow coming in on the far right of the picture? Well, that's actually what I mentioned before. It's the helper T cell that is only allowed to talk to the cytotoxic. So the helper T cell is the only one that can talk to it. Now you already have this on the original. This is actually your cytokine, your interleukin that's on your original chart where the helper T cell is talking to the cytotoxic helper cell. All right. Now in this picture, again, we see level 2 being a macrophage or level 3 being a B cell. This time it's talking to the cytotoxic and it's doing what? It's saying, hey, I need to activate the cytotoxic for the current and I need to also have memory cytotoxic for the future. So level 4 does have memory. That's page 874. Moving to the opposite page, this is now B cells. So the story about the B cells. Here we see also what? Level 4 talking to level 3. And you have a dotted line mentioning the interleukin there where 4 talks to level 3. And here we see it by way of interleukin here activating the B cells. Now these B cells can come in two forms because remember, even level 3 has memory. So we can make memory B cells for the future and we can make current plasma cells that make current antibodies. So both level 3, level 4 have memory. Okay, next slide. That was page 875. Okay, okay, moving on. And my next picture is going to be page 877. This is page 877. This is a picture of an antibody. Notice it has two major regions. The top region is the variable region. This is the region that we can adapt to whatever needs we need to make the millions, and actually I could even say gazillions of different variations of this top part. So the part that binds to the antigen, the variable region, we can make gazillions versions of those based upon our need. The bottom part is the constant region. This is the part that stays the same. So the body does not waste. It pretty much keeps this part the same for all antibodies. And the only part that changes is the antigen binding site that's different. So this is a picture of an antibody. Next slide. That was page 877. Our next picture is going to go to page 879. Now, before I continue with this picture of 879, the slides that I skipped a moment ago, those are helpful because you need them in order to make this chart. Now, this chart is valuable enough to put on a flashcard. So you should have 5 flashcards. 1, 2, 3, 4, and 5. So 5 flashcards. Now, what I'm going to do is give you a sort of a brief info of some of these. But again, the chart's very important. First one is IgG. Now, this is the most common form of antibody. And notice it's the same thing as saying what word? Immunoglobulin. So IgG is the most common form of antibody. The next version is IgM. Now, IgM is a pentamer. That means it looks like the pentagon shape. So there's 5 of these put together to give a pentagon shape. The next one is IgA. And notice it deals with mucus, saliva, tears, things like that. So that's IgA. The next one that I don't have much info for is IgD as in dog. The only thing I can tell you is IgD is a dimer. That means it's 2 of those put together, end to end. So IgD is a dimer. The last one is IgE for Edward. Now IgE is going to have a chain reaction information. So here's how that goes. IgE will deal with basophils. Yes, IgE is basophils. It's the same one that deals with allergies. So basophils. Now basophils are also related to mast cells. Now, so on our paper so far, we have IgE, basophils, and mast cells. Now, mast cells are the ones that produce histamine. So when someone has a mosquito bite and we have a swelling, that allergic reaction that you get from that, where you have a swelling, that's because of histamines. If you have an allergy to ragweed pollen and your nasal passages are swollen, that's also because of histamine. Now, when you buy Benadryl, Benadryl is an antihistamine, and that's how you curb some of the action of both the IgE, the mast cell, the basophil, because they produce a lot of histamine. So Benadryl would be the antihistamine. Okay. All right, that's page 879. Now, pages 880 and 881 are basically summaries of the pictures that I've gone through today. All right, moving on. The next very, very important page is page 883. So page 883. Now, this page will show up on a test. So this is very, very important. Now, page 883 talks about what happens first time around versus what happens the second time around. So the first time you get sick from an infection, the first thing that rises is IgM. But notice it's still a relatively small bump, so IgM. But then the major one, IgG, follows sometime after, and that's during the first infection. Now, if by chance, because of memory, remember level 3 does have memory, because of the humoral immunity of level 3, the antibodies have memory. So what happens the second time you're infected with the exact same germ? And as you can see from the slide, it says it has to be the exact same germ. The second time around, it's IgG that kicks in. And notice how fast it kicks in. It doesn't take much time. So the first time you got sick, it took a while for IgM and IgG to work. The second time around, IgG kicks in quickly, and that's what provides you the protection when you get sick from the same germ the second time around. Most often, you don't even notice it. So IgG kicks in best the second time around. So page 883, this is definitely a testable question. Okay, moving on. I'm now moving to this slide on page 883. This is where most of the classes actually stopped at. But what I want to do, just because we're Friday only, I want to make sure we sort of touch on this as our ending point for today. First thing we want to get down is what is active versus passive. So this is page 883. Active means that you yourself somehow made the antibodies. So you're making the antibodies inactive. Okay? So somehow you made them. Now, passive, which is the blue banner on the opposite side, passive means that somebody else made these antibodies. Now, when someone else makes the antibodies and they're somehow given to you, then it doesn't last as long. So passive immunity, the blue one there, passive immunity is short term. It doesn't last long. Active immunity, since you went through the trouble of actually making the antibodies and getting sick from it or whatever, the active immunity will last for decades, a very, very long time. Okay, so that takes care of the yellow and the purple at the top there in terms of active versus passive. Then the next thing, we see these four boxes at the bottom of page 883. The next thing you want to understand is natural versus artificial. Natural means that you got it into your body naturally and you end up getting sick from it or whatever it is, but it's natural. Artificial, by definition, means it's a shot. Now, I did not say vaccination. All I'm saying is that anytime it says artificial, it means it's a shot. Okay. Now, that concludes our presentation for today, ending on page 883. This is where the other classes stopped. I've got you up to that same point as well. All right. Thank you.